You know, I've been thinking, and science can be extremely hard sometimes, especially when it comes down to the way that we choose to study human diseases. You see, as scientists, we like to use animal models to better understand human disease, but animals just aren't humans. And sometimes these animals don't even develop disease in the same way that humans do. So that means that the things that we find from these animals don't necessarily help us to create the best drugs or treatments for human patients. This is especially true for diseases that don't have cures, like multiple sclerosis, or MS for short. MS is an autoimmune disease of which your immune cells will attack your brain and your spinal cord, causing damage. This results in symptoms in patients like walking difficulty, eyesight loss, pain, or even brain fog. And while there are a plethora of animal models that we can use to study multiple sclerosis, the mouse model is the most common model. However, the mouse model only develops disease in the spinal cord. So, what about the brain? How do we study the brain? This is where my research begins. Since joining the lab, we have generated humanized mouse models. These mice are missing specific mouse genes and they have an insertion of specific human genes related to the immune system, thus giving us insights into how human immune cells or human immune responses promote MS. And while these mice look like Stuart Little, they don't talk. But most importantly, these mice develop brain specific disease. And we know that this brain specific disease is also a part of MS patients, but they also develop a walking difficulty as well. So, how do these mice develop brain specific disease? A large portion of my thesis has focused back onto those immune cells. So, there are a lot of different immune cells. In the same way, there are a lot of different kinds of cars. We have Fords and Hyundais, and similarly, we have different immune cells. My research has identified one specific immune cell called the T helper 17 cell that is responsible for causing this brain specific disease. Immune cells are just like cars. They travel on a highway known as the veins to get to different tissues. While they're traveling on this highway, they use GPS signals or even road maps to guide them to specific areas like the brain. We know that once these T helper 17 cells get to the brain, that they're there to cause damage and to promote other cells causing damage. Therefore, my research has identified a humanized mouse model that develops a brain specific disease, as well as cellular targets that researchers can now use to create better treatments for MS patients. Thank you.